Hello, this is Mr. Buss, and I'm going to walk you through day one in this video of a Taste of Genetics mini lab. And so this is the equipment you'll need. Of course, you'll need 2 pipettes actually this time is helpful. One of them would be a 2 to 20 microliter, and the other one can be a 20 to 200 microliter. 2 empty, to start with, PCR tubes. These are very small little tubes. One small test tube with ES marked on it that I prepared for you. So that just is the extraction solution, the DNA extraction solution. That's going to have some liquid in it, already set for you. Sharpie, pipette tips, a container for the used tips, directions, and then this is the liquid that you'll basically be sloshing around in your mouth for a few minutes. In terms of for each group, for each sample you're going to do, so you would need 2 empty test tubes. So since I'm just running this one just for myself, I'm using 2 test tubes. If you have multiple people in the group that are running the lab with their DNA, then you'll need 2 test tubes for each person, and you'll need 1 cup of the salt water for each person as well. But then the actual instructions for you guys are going to start on page 17. So here we go. So page 17. I'm going to basically be running you through the steps, starting with step 2 because we already did the step 1 in class. I'm going to start with step 2 on page 17 in this video, and I'm going to end with step 11 on page 19. So that's where this video will take you. All right, so step 2, go ahead and label the PCR tubes. I've already done that. So I labeled them with my initials, JB, and then I put a 1 pretending I'm in like first hour. Okay, that was easy. Step 3, it says to pour the saline solution in your mouth and swish vigorously for 2 minutes. You're trying to get some cheek cells basically out of your mouth and into the cup, and then you spit back into the cup. So I'm going to go ahead and do that. Okay, that was gross, but I did it. I spit the solution back into the cup after I swished it around vigorously for 2 minutes. Now I'm going to pipette 200 microliters. So I'm going to grab the 20 to 200 milliliter pipette. It's set at 100. I need to set it to 200. All right, so this says 200, and that's a 20 to 200 microliter pipette. So we're at 200. Get a pipette tip, PCR tube, open the cap. Be careful not to squish these. They're very fragile, and if you squeeze them too hard, then they will crack. So get 200 microliters. It looks like that much, and then okay. So that is ready to be centrifuged. Okay, so here's the centrifuge. I'm going to load it. Make sure to always load it balanced. So if I'm only going to run 1, so I actually have to balance it by putting another one in there. So I actually made 2 samples for myself, but this in class this will be probably loaded with all 6. Go ahead and turn it on. And that's going to spin for 3 minutes. Okay, so it's been 3 minutes. I'm going to shut off the centrifuge. Carefully take the sample out. Okay, now what I want to do is I want to remove the liquid without removing the cell pellet. Let me see if I can focus in on the cell pellet. So right here is what I want to keep. There's a white cell pellet down there. So carefully remove the top of the tube. Hard to show in the video here. And then remove the liquid. Got it. And then there's still a cell pellet, that white cell material. And the liquid is in the pipette. I just still had it set at 200. And I'm just going to put that into the cup that I spit in originally. Okay. Now what I'm going to do is I'm going to add DNA extraction buffer, because that white pellet represents, hopefully, some cheek cells. Okay, so for this step, I'm on step 7 in case you were wondering. I'll get a new pipette tip. Always get a clean pipette tip. And it says to grab 50 microliters, and I'm still set at 200 here, so now is a good time to move this down to 50 microliters. Okay, so that's set at 50 microliters using the 20 to 200 microliter pipette. Okay, extraction solution. Remove 50 microliters of extraction solution. Add it to my tube containing the cell pellet. And then what I'm going to do is I want to pipette that up and down and slosh it around basically until it becomes resuspended. So I'm just going to kind of keep moving that back and forth until the cell pellet goes back into solution and the solution is now cloudy again. Okay, so that was an important step. Now step 8. All right, so here's the PCR machine. I'm just gonna open it and then find a spot in there, and then in class you're just gonna have to wait till we fill up all 16 of these basically. But since I'm the only one running this right now, I'm just gonna put one in there, I'm gonna close it, and then I have an app on my iPad that's going to run this. So my iPad actually runs the PCR machine, so I'm going to I set up the protocol already, and so I'm going to run. Basically, it's going to run for 5 minutes at 95 degrees Celsius. Okay, so then I'm just going to hit play on this app, and the machine is running, and looks like it's at 50 degrees Celsius, 56, 61, and so on. So as soon as it hits 95 degrees Celsius, it will start the 5-minute countdown. I'll wait a little bit since we're almost there. So now, yep, so it reached 95 degrees Celsius, and now it's going to run for 5 minutes, and we're breaking apart the cells. 95 Celsius is pretty hot. Remember, water boils at 100, so we are near boiling temperature already. Okay, so the app that runs the PCR machine is saying that it's done, and it is now holding a temperature at 4 degrees Celsius. So it ran 95 degrees Celsius to break apart the cells for 5 minutes, and now it's holding it at a cool temperature. And so what we're going to do now is we're going to remove the DNA from the tube. Remove my DNA sample. Okay, and what I'm going to do, let me zoom in on that, is I'm going to pipette 5 microliters out of here and into a new tube. Then I'm going to throw this away. Okay, so I've got my pipette. Since it's a small volume, I've got my 2 to 20, because 5 is within 2 to 20. I've got this set at 5.0. Going to open up the cap of the new one. Open up the cap of the one that I just had running in the PCR machine. And then I'm going to just carefully pipette 5 microliters out of here. It's not very much. 5 microliters of my sample, and then I'm going to freeze that immediately. So that's going to go right in the freezer, and then this is ready to start day 2.